So, good morning, everybody. We had discussed about a general view about introduction and mechanism of urine formation in the last class. Today, we'll have a detailed discussion regarding this. So, mechanism of urine formation includes GFR, tubular secretion, and tubular reabsorption. What is that which has to be excreted? The major metabolic wastes produced by humans are water, carbon dioxide, salts, and urea. So, this depends on the dehydration, synthesis, and respiration. So, how water, if it is in a winter, you pass more urine, or during cellular respiration, you liberate the metabolic end products, carbon dioxide, or the neutralizing reaction results in salt formation. Protein metabolism and deamination can result in formation of urea and nitrogenous wastes. So, urea in turn forms amino acids where ammonia is removed in the form of ammonia and liver converts into urea. And with nucleic acid catabolism, there is formation of uric acid, and creatinine phosphate results in formation of creatinine. So, in today's class, we'll be dealing more about the urine formation. So, we need to encompass what is the basic filtering membrane. As you all know, the peritubular capillaries is present in the cortical nephrons. It arises from the efferent arteriole. It's low pressure. Capillaries attached to the venule. It clings to the renal tubule. It reabsorbs substances from the collecting tubes. So, the urine formation includes three processes. One, the filtration, which is the ultrafiltration, the reabsorption, which is the tubular reabsorption, and tubular secretion. So, the preview of the urine formation is, as you all know, the glomerular filtration creates a plasma-like filtrate of the blood. There's no plasma proteins in it, whereas tubular reabsorption removes useful solutes from filtrate and returns them to the blood. The tubular secretion, predominantly hydrogen and potassium, removes additional waste from the blood and adds them to filtrate. And finally, how the water is conserved, either by facultative and obligatory reabsorption. Obligatory takes place in the proximal convoluted tubule due to the osmotic gradient created by the sodium, resulting in absorption of water, whereas the DCT results in the presence of antidiuretic hormone through the water channels, that is aquaporins, water is reabsorbed. There are three steps in urine formation. Once again, in glomerular filtration, the water and solutes in the blood plasma are filtered out of the glomerular capillaries. The water, ions, nutrients are selectively reabsorbed from glomerular filtrate and returned to the blood in the peritubular capillary. That is what you saw in the first line, that is the tubular reabsorption. The tubular secretion removes waste products and substances in the excess from the blood in the peritubular capillaries and into the renal tubule. So, what is urine? It's nothing but glomerular filtration plus tubular secretion plus tubular reabsorption. So, glomerular filtration, you all know the rate at which the filtrate is formed in all the nephrons in a unit time. The normal GFR is 125 ml per minute and it is 180 liters per day. And you all know during the process of filtration, a protein-free and a cell-free filtrate is formed in the PCT, hence it is ultrafiltration. The filtrate is called ultrafiltrate. The filtrate resembles plasma except for the absence of proteins. It's filtered through the fenestrated epithelium. About 180 liters of plasma are filtered and they form the plasma. The filtrate, the glomerular filtrate. The filtrate is nothing but the plasma without the proteins, and about 2 liters of urine are produced per day. It's efficient because of the fenestrated glomerular capillaries, which prevents the loss of large plasma proteins, which has molecular weight more than 60,000. That's the reason the albumin is not filtered. If albumin is filtered, it indicates the onset of renal failure and blood cells. The particles from ferrocytes reduce the size of fenestrations. The filtration slit and which prevents the loss of medium-sized plasma proteins. The non-selective process allows solutes such as ions, glucose, urea, ammonia, vitamins and amino acids from the blood to pass, which in turn prevents the filtration of large plasma proteins such as albumin, blood cells and platelets. So what's filtration? Pressure. Use of pressure to force fluids and solutes through the membrane. The three reasons for large volume through the renal corpuscles are the large surface area of the glomerular capillaries, the thin porous filtration membrane. The diameter of efferent arteriole is larger than the efferent arteriole. The high glomerular capillary blood pressure of 55 or 60 millimeters of mercury. This all enhances all these three factors: surface area, the porous filtration membrane and the diameter, all enhances the filtration pressure. So filtration pressure is responsible for filtrate to be formed. The glomerular capillary pressure minus the capsular pressure minus the osmotic colloidal pressure forms the filtration pressure. The changes caused by glomerular capillary pressure, and you all know the values. The glomerular capillary blood hydrostatic pressure is 55, whereas the capillary hydrostatic pressure is pericapillary, it's around 15, whereas the colloidal osmotic pressure, it's always constant at 25. So if you can calculate the glomerular blood hydrostatic pressure, subtracting the colloidal hydrostatic with the Bowman's capsular colloidal osmotic pressure, so the filtration pressure is around 10 millimeters of mercury. So this is very important. So what are the forces acting on the filtration? The glomerular capillary hydrostatic pressure results due to the hydrostatic pressure against the capillary wall. It's around 55 to 60. And you all know that the glomerular capillary hydrostatic pressure. Filtration is favored by capillary hydrostatic pressure. An increase in capillary hydrostatic pressure enhances the effective filtration pressure and hence filtration takes place. So this capillary hydrostatic pressure promotes filtration. The Bowman's capsular colloidal osmotic pressure due to the presence of the solutes, it's around 0, and this also promotes filtration. The forces acting on filtration. So we have two forces which promotes filtration and the forces which oppose filtration are the glomerular osmotic pressure due to the presence of proteins in the blood, it's around 25. The filtration is opposed. There is no filtration taking place, due to which the RBC and albumins in the capillaries are not filtered. The Bowman's capsule, the colloidal osmotic pressure, it's a pressure excited by plasma protein colloids and it opposes outward movement of the fluid. This force tries to retain the fluid within the blood vessels. Whereas the Bowman's capsular hydrostatic pressure exerted by the fluid present in the Bowman's capsule, this also opposes filtration of the fluid at the glomerulus. So we need to know that the hydrostatic pressure is 45, colloidal is 25 and the Bowman's capsular pressure is 10. All these put together we get the effective filtration pressure. We're subtracting colloidal osmotic pressure and Bowman's pressure to the hydrostatic, we get around 10 millimeters of mercury. So this is how you calculate. The fluid moves from the glomerulus to the capsule, Bowman's capsule. So this is the net filtration rate. So filtration rate and filtration fraction we need to understand. The glomerular filtration rate, the volume of plasma filtered per unit time, that's around 125 ml per minute or 180 liters per day. So filtration fraction is glomerular filtration rate divided by renal plasma flow, which is around 20%. So there's another thing called as filtration coefficient, which measures the conductivity and surface area of the glomerular capillaries. So the coefficient is equal to filtration rate by filtration pressure. It's around 12.5 ml per minute per millimeters of mercury of filtration pressure. So what are the factors affecting GFR? So glomerular filtration rate is directly related to the filtration pressure. The glomerular capillary is located between the afferent and efferent arterioles. Any change in the diameter of these will affect the pressure, and this will change the GFR. So amount of filtrate produced in both the kidneys per minute. If the GFR is too high, the needed substance cannot be reabsorbed quickly enough and are lost in the urine. Whereas if the GFR is too low, everything is reabsorbed, including waste that are normally disposed of. So how is GFR regulated or controlled? It is by three mechanisms. One is the autoregulation. The definition of autoregulation: ability to regulate its own blood flow despite variation in perfusion pressure is called as autoregulation, wherein it's constant between a pressure of 60 to 160 millimeters of mercury, and this renal autoregulation is dependent on two mechanisms: the myogenic mechanism and tubular glomerular feedback. Whereas the neural regulation is dependent on the sympathetic and parasympathetic, which is the epinephrine and norepinephrine, whereas the hormone regulation is the renin-angiotensin aldosterone mechanism and the atrial natriuretic peptide. So if you take the myogenic theory of the autoregulation, increase in blood pressure enhances the flow of blood onto an organ. So this is mainly because of the myogenic tone with the sympathetic vasoconstrictor tone, which fires at a rate of 1 impulse per second. The ability of the kidney to maintain GFR without nervous and hormonal control. So myogenic mechanism is dependent on juxtaglomerular cells, whereas the tubular glomerular feedback is by macula densa, which is a sodium sensor. So when systemic BP rises, walls of the afferent arteriole are stretched. The JG cells surrounding the afferent arteriole contract, wherein the diameter of the afferent arteriole narrows down, and thereby the renal blood flow slows and GFR drops. This is due when the blood pressure decreases. That's the reason we need to maintain a perfusion pressure of around 90 millimeters of mercury, or otherwise there is renal failure setting in due to diarrhea or any cause which can be primary or secondary. When the systemic blood pressure falls, the afferent arteriole relaxes and the diameter increases. Thus, the blood flow remains stable. So, another mechanism, which is the tubular glomerular feedback. The DCD provides a feedback to the glomerulus, that's a distal convoluted tubule. When the systemic blood pressure rises, the glomerular filtrate moves rapidly through the renal tubule, thereby resulting in reabsorption of sodium chloride and water, which slows down. The macula densa cells in the GCT sense the concentrated filtrate and stimulate the JG cells. The JG cells constrict and the GFR drops to normal. So the next, we came across the factors influencing GFR and how is it regulated. The next process which involves in the urine formation is tubular reabsorption, wherein it's a process where most tubule contents are returned to the blood. That's the peritubular capillaries and the cortical nephrons. The transported substance is moved from the tubule to the peritubular capillaries. The solutes are reabsorbed by active or passive transport mechanisms. So how do we go about this? The proximal convoluted tubule reabsorbs almost 63 to 65% of the filtrate, and it returns back to the capillaries. Here it's the sodium chloride, glucose, amino acids, water, urea, uric acid, hydrogen ions, potassium, magnesium, calcium, vitamins, and lactic acid all are reabsorbed back into the capillaries. This is assisted by the active transport or with the help of antiport, facilitated diffusion, simple diffusion, and pinocytosis. These are the transport mechanics. Creatinine, sulfates, phosphates, nitrates are not reabsorbed. The reabsorption is limited by a number of transport proteins in the plasma membrane. Glucose appearing in the urine is called as glucosuria. More glucose in filtrate can be reabsorbed by tubule is equal to glucose in urine. More glucose in the filtrate that can be reabsorbed by tubule can result in equal glucose in urine. If it is more glucose, it results in glycosuria causing diabetes mellitus depending on the threshold. So peritubular capillaries are specialized for reabsorption because the colloidal osmotic pressure is more than the Bowman cytostatic pressure in the peritubular capillaries. The walls of the capillaries are permeable. Reabsorption occurs throughout the renal tubule, which can be active or passive process. 99% of filtered water is reabsorbed in the blood and returned to the body. So reabsorption needs peritubular capillaries, which assist in the reabsorption of water, glucose, and amino acids and ions. Some are by passive process, some are by active process. Most of the reabsorption occurs in proximal convoluted tubule. The substances which are not reabsorbed are the nitrogenous waste products like urea, uric acid, and creatinine, and excess water. So the next process in the formation of urine is the tubular secretion, which is about the secretion of hydrogen ion and ammonia, which plays a role in acid-base balance with the hydrogen ion, secretion of potassium, secretion of creatinine, secretion of certain exogenous substances such as diuretic, mercurial diuretics, and penicillin. So what's this tubular secretion? Chemicals from the capillaries which were reabsorbed or newly formed chemicals are secreted in the tubular fluid. This happens in the proximal convoluted tubule and the loop of Henle. The waste which is removed, that's urea, uric acid, bile salts, ammonia, and catecholamines and creatinine. Some drugs like penicillin are secreted. This tubular secretion helps in acid by maintaining the acid-base balance. When hydrogen ions and bicarbonate ions regulate the pH of the body fluids. Secretion and reabsorption are in reverse processes. Materials move from peritubular capillaries to renal tubules, whereas from tubule to the peritubular is the reabsorption. The opposite of it is the secretion. The substances which are secreted are hydrogen, potassium, creatinine. Materials left in renal tubule move to the ureter. So reabsorption and secretion of the kidney involve the transport mechanisms like the passive, which involves diffusion and osmosis, channel mediated diffusion, or carrier mediated transport, which is facilitated. The transport of ions is driven by two general mechanisms: the active transport in an energy which is dependent on ATP, utilizing it, whereby ions are pumped against the electrical chemical gradient by the uphill gradient, or passively through the electrical chemical gradient, which is downhill along the gradient created by active transport. The routes of reabsorption can be carrier mediated, which involves the facilitated, the active, the co-transport, and the counter-transport. The 5 characteristics of carrier mediated transport are: specific substrate binds to carrier protein, which facilitates movement to cross the membrane. A given carrier protein usually works in one direction only, and the distribution of carrier proteins varies among proteins of cell surface. The membrane of single tubule cells contains many types of carrier protein. Carrier proteins like enzymes can be saturated. The ions are transported, particularly the sodium from the lumen to the blood across the tubular wall, is through transcellular and paracellular pathways. The transcellular pathway is through the apical surface of the epithelial cells of the PCT. The paracellular is between the epithelial cells of PCT. Paracellular is passively by solvent drag. Sodium can also move passively into the proximal convoluted tubule via the solvent drag, where the movement of water driven back to sodium transport sweeps additional sodium and chloride along with it, which is mainly due to a sort of mass action of the lumen into the lateral intercellular space. The paracellular movement can also be due to the facilitating passive transport, which is leakiness of the nephron which is maximum in the proximal convoluted tubule and decreases along the nephron to the collecting ducts. The transcellular can be by luminal stubs, which involves diffusion mediated by co-transport or by exchange by primary ADP driven pumps like halogen potassium or the sodium potassium ATPs. Transcellular pathways are mainly through the basolateral, which are by facilitated carriers, co-transport or the exchanges, and passive diffusion. The 5 functions of the proximal convoluted tubule is about reabsorption of organic nutrients, active reabsorption of ions, reabsorption of water, passive reabsorption of ions, and secretion. So reabsorption, which takes place in the proximal convoluted tubule, is predominantly where the glucose, amino acid, sodium will be pumped out of the tubules by active transport, wherein it is ATP dependent, whereas the chloride will follow the sodium into the peritubular capillaries space. It's accumulation of positive charges resulting in drawing of the chloride outside the tubule. Water will pump into the peritubular space because of osmosis, which is due to the osmotic gradient created by the sodium. Some compounds are present in high concentration in the filtrate, but low in the blood can move through the diffusion. So glucose reabsorption also relies upon sodium gradient. Most is reabsorbed in the proximal convoluted tubule. All the glucose that is at apical membrane needs sodium glucose co-transporter. All the glucose that is filtered gets reabsorbed by co-transporter secondary active transport with sodium ions. Energy for transport is provided by sodium potassium AD base. So at the apical membrane it needs sodium glucose co-transporter crosses the basolateral membrane via the glut, which do not rely on sodium. So there are 2 transporters, sodium glucose transporter and glucose transporter. From the lumen to the tubule is by SGLT. From the tubule to the capillaries is by glut. So we need to understand this. So glucose is reabsorbed by proximal convoluted tubule. No glucose appears in the urine. There will be absence of glucose in urine. The proximal glucose transport is actively coupled to sodium transport via sodium glucose co-transporter on the apical membrane and across the basolateral membrane by facilitated diffuse depending on the glucose transporter 1 and 2. And the early proximal is driven by transporter SGLT2, but later the proximal along the proximal by SGLT1. So the amino acids are reabsorbed totally. The apical amino acid transporter, the active transporter, sodium dependent co-transporter, though some amino acids are reabsorbed via sodium independent facilitated diffusion. Or on the basolateral, amino acids exit by facilitated diffusion. Amino acid transport is similar to glucose in that they exhibit transport maximum and may saturate if plasma levels are too high. Reabsorption is to preserve the essential nutrients for the energy. The tubular maximum values are lower than that of glucose, so can excrete excess in the urine. Amino acid transporters rely on sodium gradient of the apical membrane, but a couple of exceptions are reabsorbed via sodium independent facilitated diffusion. It also exits through the basolateral membrane via diffusion, but again, some exceptions rely on sodium. The tubular maximum of a substance is that for most substances that are actively reabsorbed or secreted, there is a limit to the rate at which the solute can be transported, which is referred to as tubular maximum. They also exhibit threshold. For example, we'll come to know in glucose, there is a renal threshold, a plasma level after which the solute starts appearing in the urine. So the renal threshold is 180, whereas the tubular maximum in males was 375 milligram per minute and 325 milligram. Whereas the threshold, if it is more than 180, it starts appearing in the urine for glucose. So the Tm, that's a tubular maximum. If a nutrient concentration rises in the tubular fluid, the reabsorption rates increases until carrier proteins are saturated. When saturated, some material will remain in the tubular fluid and appear. Tubular maximum determines the renal threshold. The substances which are not reabsorbed, the tubular maximum is different. It reflects the number of tubular maximum, reflects the number of carriers in the renal tubules available. It exists for nearly every substance that is actively reabsorbed. When the carriers are saturated, excess of that substance is excreted. So this is how the glucose starts appearing in the urine. We had discussed when all the nephrons do not have the same capacity of reabsorption in an ideal nephron. All the nephrons are not active and absorbing to the maximum. All the maximum capacity of reabsorption of glucose is to be 325 to 375. The actual reabsorption far below the expected level. This deviation of actual absorption compared to maximum is called a sleeve, which is rounded. So maximum rate at which substance can be actively absorbed. Each substance has its own tubular maximum. So glucose, amino acids, phosphate, sulfate, uric acid, diuretics and salicylic acid all are examples showing tubular maximum. The tubular maximum for glucose is 320 mg per deciliter, whereas the filtered load of glucose is only 125 mg per minute, that is 1 mg per ml into 125. Above tubular load of 220, it begins to appear in the urine. So glucose in the blood is tightly regulated by insulin to around 70 to 100 mg per deciliter. The transport of a glucose on the basolateral membrane is the GLUT has a limited capacity to carry glucose back into the blood. If blood glucose rises above 180, some of the glucose fails to get reabsorbed and remains in the urine, and this is called as glycosuria. So reabsorption almost takes 90% of the proximal convoluted tubule via passive transport, active transport, co-transport, specialization of tubule segments. The distal tubule and the collecting tubule are affected by hormones like ADH and aldosterone, which enhances the sodium reabsorption, and the ADH enhances the water reabsorption. The substances transported or active transport moves sodium across the nephron wall. Other ions and molecules moved by co-transport. The passive transport moves water, urea, lipid soluble and non-polar compounds. The substances for tubular reabsorption and secretion, where you can see that most of the substances are reabsorbed in the proximal convoluted tubule, and some of the substances like hydrogen and potassium are secreted. So today's class we saw about how the urine formation takes place, the mechanism, which involves three processes called as GFR, tubular reabsorption and tubular secretion. You know the amount of filtrate found by all the nephrons per unit time, which is the GFR. It's around 125 ml per minute, and tubular reabsorption takes place maximum in the proximal cannulated tubule depending on the transport mechanics. For example, most of them, and along with sodium, water is reabsorbed due to the osmotic gradient, whereas in the distal cannulated tubule, the water is reabsorbed due to the presence of ADH. So, you know the regulation of GFR and what is tubular maximum and what is play. So, in spite of the renal threshold being 220, 325, because all the nephrons do not have the same capacity of reabsorption, and all the nephrons are not active and absorbing to their maximum at any given point of time. Thank you. Next class we'll be talking about countercurrent mechanism, how water is concentrated in the loop of Henle in the juxtamedullary nephron. Thank you.